It is easy to picture a spinal cord injury as a single, devastating moment. The bone breaks, the cord is pinched, and a vital line of communication snaps like an electrical wire. But the initial impact, the primary injury, rarely severs the entire cord. Instead, that physical trauma acts as a detonator for a prolonged secondary disaster. Within minutes of the accident, surrounding blood vessels rupture, starving the surviving tissue of oxygen. Cells burst, flooding the local environment with toxic levels of calcium, which triggers a wave of cell death. Then the immune system makes a fatal miscalculation. T cells, designed to fight infection, arrive at the trauma site. They misidentify myelin-basic protein, the protective coating around nerve fibers, as a foreign invader and latch onto it. The immune cells strip this myelin away from the surviving nerves. Without that insulation, the axons are left entirely exposed and unable to transmit electrical signals. To contain the spreading damage, local cells called astrocytes swarm the area. They multiply and weave themselves together, forming a dense web of fibrous tissue known as a glial scar. This scar acts as a biological brick wall. It physically blocks severed nerves from growing back together and secretes chemical inhibitors that repel any attempting to cross. The paralysis that follows is rarely caused entirely by the initial impact. It is locked in place by this hostile, self-inflicted microenvironment. This diagram illustrates the timeline of the secondary injury cascade. By intercepting this process during the acute phase, the first 0 to 2 days, researchers can alter the long-term biological outcome. One method is immunization with neural-derived peptides. Instead of shutting down the immune system entirely, which would leave the body vulnerable, these synthetic peptides modulate it. The peptides induce a state of protective autoimmunity. They convert aggressive inflammatory M1 macrophages into healing M2 macrophages, which begin releasing neurotrophic factors that protect the surviving tissue. Another chemical intervention uses a peptide known as PR21. This molecule mimics polysialic acid, a sugar naturally present in the developing brain that guides new nerve growth. This chart details the concentration of PR21 when delivered directly to the lesion site in a mouse model following a spinal cord injury. The dashed line represents the 40 micromolar threshold, the minimum concentration required for the peptide to be biologically effective. At both 24 and 48 hours post-injury, the local concentration of PR21 remains well above that necessary level. By deploying these peptides in the critical window after an injury, the secondary damage cascade is suppressed, leaving a tissue environment that is capable of accepting further repairs. Once the initial inflammation subsides, the chronic phase begins. The injury site is now characterized by a physical dead zone, a cystic cavity devoid of the tissue needed to transmit signals. To bridge this gap, engineers synthesized porous, three-dimensional biomaterial scaffolds made from molecules like hyaluronic acid or chitosane to physically fill the cavity. These scaffolds are seeded with neural stem cells before implantation. The structure gives the cells a physical substrate to cling to, allowing them to differentiate and grow new axons directly across the dead zone. Reconnecting the biological wires is only half the solution. The muscles have atrophied from disuse, and the newly formed neural circuits require training to coordinate movement. This illustration shows a functional electrical stimulation, or FES ergometer. By applying targeted electrical currents to specific muscle groups, FES forces paralyzed limbs to contract and cycle through continuous pedaling motions. This schematic demonstrates an implantable FES chip wired to the perineal nerve of a rat with a completely transected spinal cord. The stimulation produces reciprocal, gate-like movements in the hind limbs, effectively forcing the repaired central nervous system to relearn motor patterns. Stem cells and biomaterials lay down the biological track, while electrical engineering provides the active stimulus required to bring those circuits online. Decades of research prove there is no single molecule or surgical technique that can cure paralysis. Restoration relies on a carefully timed combination of interventions. It begins with chemical peptides, delivered within hours of the injury to suppress the runaway immune response and halt the spread of secondary tissue damage. Next, physical scaffolds and neural stem cells are implanted to bridge the lesion cavity. laying down a matrix of new axons through the injury site. Finally, advanced bionics and electrical stimulation are applied to force movement, training the new neural connections to fire in coordinated patterns. By layering these targeted treatments, we are moving toward a reality where severe spinal cord trauma is treated as a manageable biological obstacle, rather than a permanent state. This exploration of neurorestoration was made possible by Synestex Systems LLC. Founded by Shane Persinder, Synestex is dedicated to illuminating the cutting edge of biotechnology and human potential. To learn more, check out the links in the description below.